Let's start with the mechanism of excitation. In the case of cardiac muscles, we have pacemaker cells, which I will discuss in a separate slide. Electrical activity of muscle cells. In the case of the cardiac muscle, we have action potentials and plateaus. The calcium sensor is troponin C. Excitation-contraction coupling, we need calcium entry from the extracellular space and calcium release from the sarcoplasmic reticulum. Extracellular space accounts for 20% of calcium, 80% comes from the sarcoplasmic reticulum. Termination of contraction. This is done by repolarization of the action potential and reuptake of calcium. Duration of the action potential and contraction up to 400 milliseconds. Regulation of force. In the case of the cardiac muscle, the force depends directly on the amount of calcium. I have discussed all these properties in details in this video. The source of metabolism in the cardiac muscle is oxidative. Okay, now let's dive into the details. The cardiac muscle can be considered as syncytium. It means once one cell has generated an action potential, it can spread it to millions of cells. We see here cardiac muscle cells, and here we see the intercalated discs. Let's blow this up, and what we see here is the following. These are the intercalated discs. We see the desmosomes here, and they have many physiological roles. Some of the most important roles are to stabilize the cardiac muscle cells mechanically and also to hold them together during the contraction of the cardiac muscle. And we also see here the gap junctions, which I have discussed in a previous part. Okay, so the current or the action potential, once generated in one cell, it spreads from one cell to the other. So this is where we need the electrical synapses, the gap junctions. In summary, the cardiac muscle cells generate action potentials. These are the pacemaker cells, which I will discuss in the next slide. And the action potentials spread instantaneously. So this is why we call the cardiac muscle cell syncytium. Here we see the cardiac conduction system. The cardiac conduction system is self-excitatory. We have so-called pacemaker cells. The sinoatrial node is the primary here. Okay, the sinoatrial node, the SA node, generates the first action potential, and the action potential is spread through these fibers. These are the pacemaker cells. Okay, and then it arrives at some point the atria, the right atrium, the left atrium. Then the conduction is slowed down by the action of the atrioventricular node here, and then it goes very rapidly to the His bundle and then to the right and left bundles. Finally, the Purkinje fibers, and this results in contraction of the ventricles. I will discuss this in details in a separate part. Okay, here we also see the ECG curve, which I will also discuss in details in a separate part. Just to briefly show you, here we have the so-called P wave, which describes the contraction of both atria, the right and left atrium. At this point here, the so-called PQ interval, this is where let me go back up here. So we have here the AV node. So this AV node slows down the conduction. Why? Because we would like to have the atria contracting first and then the ventricles. The AV node will slow down the conduction. So then we have the QRS complex. This is the contraction of the ventricles. And then we have the T wave, which is the repolarization of the ventricles. Again, I'll discuss this in details in a separate part. The cardiac action potentials vary strongly. You can see here an action potential of the pacemaker cells, SA node, okay? No resting memory potential. It's quite unstable. And this is the normal cardiac action potential in the myocardium, for instance, okay? Remember, this here here, 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 the plateau phase, which I discussed in the previous part. This is where we have calcium influx. And this is where we also have the contraction of the cardiac muscle cells. The cardiac muscle is subject to autonomic control, sympathetic and parasympathetic systems. The sympathetic system activates or erases the cardiac activity. So it can increase the heart rate, can increase the speed of contraction. It can also increase the force of contraction. And exactly the opposite effects are done by the parasympathetic system here, okay, and here. So the excitation and contraction coupling and the sources of calcium, remember I discussed this in the previous part, and I had a summary for you here. So just to briefly summarize, the sources of calcium are again 20% from the extracellular space, 80% from the sarcoplasmic reticulum. And then we have the cross-bridge cycle. At the end, we need to bring calcium back into the sarcoplasmic reticulum and out of the cells. So this is the repolarization of the cardiac muscle, which I discussed in the previous part. Okay. The cardiac muscle cells cannot exhibit tetanus. Also this, I will deal with in details in a separate part. Just to briefly introduce it, tetanus is the following. This here is a skeletal muscle. One contraction is one twitch. Assume we would like to have one twitch after the other, say with a high frequency of activation. This would be the first activation, the second, the third, the fourth. If the frequency of activation is high enough, all of these twitches will fuse and we have a so-called tetanus here. Okay? High stimulation frequency leads to tetanus. This is possible in the case of skeletal muscle cells because the action potentials there are very short as compared to the action potentials in the cardiac muscle. It is not possible to have a tetanus in the cardiac muscle because remember here, if the SA node triggers the next action potential, okay, as it arrives in myocardial cells, it arrives at this point here, which is an absolute refractory period. It's impossible to answer the next action potential if it lands here. Okay? I have a summary here for you. The myocardium cannot exhibit summation of tetanus due to its long refractory period, lack of motor unit recruitment, and intrinsic rhythmicity. These characteristics, okay, ensure optimal heart function and prevent harmful conditions. This too I'll discuss in details in a separate part. The sources of energy in cardiac muscle cells. Oxidative energy sources. So, I have a summary here. Healthy myocardium uses oxidative energy sources, mainly fatty acids, up to 70%, 65 to 70%, with little contribution of glucose. Okay? However, the heart can also utilize lactate, ketone bodies, amino acids, or even acetate. It can adapt to the physiological and pathophysiological conditions. I hope this was helpful.